Good afternoon. I'll begin by discussing the epidemiology of snakebite and the clinical presentations of envenoming, and then we'll talk more about the treatment of snakebite and the current situation we're facing regarding antivenom availability in Africa. Published data about the burden of snakebite are limited and of variable quality. Like many other neglected conditions, estimations about the burden of disease vary widely, but it likely affects millions every year and causes tens to even hundreds of thousands of deaths. One of the reasons that the data is notoriously bad is that snakebite disproportionately affects the rural poor. Antivenoms are often not available, and snakebite victims know that. So they care for their bites at home, hoping for the best, and they don't come to seek medical care, so we can't count them. So one way to get around this problem is to do community-based surveys. One such survey was carried out by Epicentre in 2015 in Agok, South Sudan, where MSF treats about 200 snakebites a year. The annual incidence of snakebite was estimated at up to 3.6% of the population per year. And to put that in perspective, that would mean that between 10 and 15 people in this room would be bitten by a snake every year. Multiple species were identified in the area, which correlates well with the clinical syndromes seen at the hospital. And many victims sought care, but not all of them, and almost everyone tried traditional methods before coming to the hospital. So this type of survey is much needed as it provides valuable insight on local practices and epidemiology. In Paoua, the Central African Republic, MSF treats about 500 bites per year. And one retrospective study recently described the clinical presentations of the cases, and nearly all of them were due to a single species of viper. A second retrospective study described a large increase in case fatality when a new antivenom, called Antivenom, was used during a stockout of FAV-Afrique. But before we go too much further, I want to outline why, besides issues surrounding access to care, the treatment of snakebite is challenging. First, the species responsible for the bite is often not known or is misidentified. And while the majority of snakes aren't venomous, for those that are, it's important to understand that venom is not a single substance. It's a mixture of proteins and enzymes with different mechanisms of action. And it's logical that different species of snakes produce different kinds of venom, but there can also be considerable variation in the venoms produced even among individual members of the same species. And this becomes very important when it comes to producing antivenoms. And then each bite is different, with a different amount of venom injected each time. And the goal of treatment is to give enough antivenom to neutralize this quantity of venom that's been injected, which is an absolute quantity and independent of the patient itself, whether they weigh 2 kilograms or 200 kilograms. And then side effects are also common: anaphylaxis in the immediate term and serum sickness over the longer term. And they vary considerably depending on the antivenom used. And finally, 3 different clinical syndromes exist. The first is hematotoxicity. And in Africa, it's mostly associated with vipers, like the saw-scaled carpet viper seen on the left, which was responsible for over 90% of the envenomings in Paoua. The venom impairs coagulability, which causes bleeding from the bite site or from the mucous membranes. An untreated case fatality can be up to 10% and happens as a result of hemorrhage, often days after the bite. Now, the neurotoxic syndromes are the ones that are most feared because they're associated with the cobras and the mambas, like the black mamba seen on the right. Neurotoxic venoms block transmission at the neuromuscular junctions. So they lead to anticholinergic syndromes, so think ptosis and drooling, and then eventually paralysis, which can happen in hours after the bite, leading to an extremely high case fatality ratio. And then the third syndrome is the cytotoxic syndrome. And the common culprits in Africa include the spitting cobras and the puff adder, which between the two are responsible for a majority of bites seen in Agok. Blistering happens very quickly and without proper care develops into necrosis, infection, and gangrene, which can be fatal. So beyond these clinical problems I've described, there's also a problem with antivenom, which is based largely on 19th century technology. Venom is harvested from a snake and then minuscule doses are injected into horses or sheep. And after a few weeks, the horse's plasma, which now contains antibodies against the different toxins found in the venom, is collected and purified. Antivenoms produced can be specific to a single snake or polyvalent. And the only new part of the process today is the purification of the sera. Removing extraneous proteins greatly reduces the risk of anaphylaxis and late-onset serum sickness. But as you can imagine, this process is lengthy and leads to a process which is very expensive. For many years, MSF has used FAV-Afrique as a standard treatment in its projects. It's a polyvalent antivenom against 10 species of snakes, and it covers all 3 of the clinical syndromes. Its production was stopped in 2013, and the last vials produced will expire at the end of this month. Now, other antivenoms exist. We'd prefer to base decisions on which ones to use based on comparative evidence, but this really doesn't exist, which is remarkable considering the burden of disease. One of the problems is that there are few researchers working on the subject in Africa, and experts in the field tend to be associated with specific antivenoms, raising concerns about competing interests. And although progress is being made, regulatory frameworks surrounding antivenoms is unclear. So one possibility is a South African polyvalent antivenom, which has been produced for over 30 years. But its continued use is based largely on subjective evidence and subjective experience with its efficacy. Very few clinical data have ever been published. One small case series from 1998 showed that 13 of 17 patients treated had anaphylactoid reactions after its use, but we're unaware of any follow-up data on this. Now, it's highly unlikely that this is still the case, otherwise it still wouldn't be widely used. So either the product was changed or there was a specific manufacturing concern at the time. But this sort of open question wouldn't be tolerated in other diseases. A newer antivenom is Equitab Plus. It's the only antivenom to have been used in a randomized controlled trial in Africa, where it showed excellent results for carpet fiber bendings in northeastern Nigeria. But it doesn't cover the neurotoxic syndromes. On the other hand, this trial compared Equitab Plus with a similar antivenom from the same manufacturer. And while there were no deaths observed, there was a high rate of immediate reactions. The newest addition is Enoserp. Its effectiveness was described in one multicentric observational study in Guinea and Benin, though protocol violations relating to dosing were reported in 60% of patients included in Benin, and documentation of safety was not listed as an objective in the published data. But it does have the additional benefit of having a thermostable lyophilized form. On the other hand, it's the follow-up product to Antivenom, the antivenom used briefly and very unsuccessfully in Paoua. Its producers claim vast changes to the manufacturing process, but there's little publicly available data. And it's a bit tricky to follow as they've recently incorporated a new Europe-based holding company while keeping their production facilities in Mexico. So, like with many other neglected conditions, this lack of data is unacceptable. But without FAV-Afrique, we're going to have to use these antivenoms, even though all of them come with their own problems. We need comparative data on antivenoms produced by different manufacturers. And given the comparatively low-tech production process that relies on harvesting venoms from different snakes caught we don't know where, we also need to ensure that we test different lots of antivenoms in multiple sites, as their efficacy might not be the same everywhere. And finally, there's a need to standardize endpoints across studies for all 3 major clinical syndromes. So in Paoua, we're trying to do the right thing, to do a study that wouldn't be met with the critiques that I've just outlined. We're attempting to conduct a randomized, double-blind, non-inferiority trial of Equitab Plus versus FAV-Afrique, which, to our knowledge, would be the first comparative trial of 2 antivenoms from different manufacturers in Africa. The real difficulty in performing this kind of trial doesn't lie in its design, but rather in the unfamiliarity with the subject among decision makers and the difficult local context in the CAR. And despite approval of ethics committees in 3 countries, including in the CAR in January, we're still awaiting approval from the Ministry of Health to begin inclusions in this trial. In Agok, South Sudan, where we've shown the heterogeneity of clinical envenoming syndromes, we'll be putting into place a prospective observational study during the rollout of the successor antivenoms to FAV-Afrique. And given this particular epidemiology, multiple antivenoms are necessary. A syndromic approach will be used for treatment, with Equitab Plus for cytotoxic and hematotoxic syndromes, and the South African polyvalent for neurotoxic syndromes. Data collection tools in Agok will be similar to those we use in the Paoua trial, and we hope that even when comparative studies aren't possible, that standardizing data collection among observational studies will bring an added value. So to conclude, snakebite is a neglected condition and a major problem in Africa, and for many reasons, it's not been a priority for ministries of health or for manufacturers. And with the studies I've described and potentially others to come, we hope to contribute to the knowledge base. It's going to be important to document our experiences with new antivenoms in MSF programs. Without better data, we won't be able to make informed decisions on how best to treat our patients and be forced to rely only on expert opinion. Thanks for your attention.
Thank you very much, Matt.